Good day to you all. Allow me to start by thanking the conference organizers for giving me this opportunity to present this work on behalf of my colleagues on the frequencies and correlates of endocervical regulatory T cells among us female sex workers in Nairobi, Kenya. Inflammation in the female genital tract, defined by elevated levels of pro-inflammatory cytokines, has been associated with an increase in HIV acquisition risk. We have also shown in a large cohort study, which recruited over 700 young women from Durban, South Africa, that inflammation undermines the efficacy of tenofovir gel in preventing HIV infection. However, when we conducted a prospective analysis of these data, we found that cervicovaginal IL-2 concentrations were associated with a reduction in HIV acquisition and low levels of inflammation in the cervix. Following up on IL-2, previous studies have shown that low-dose IL-2 infusions in healthy individuals promote the function of regulatory T cells. Furthermore, low-dose IL-2 also mediates clinical improvements in autoimmune and inflammatory disease conditions. This led us to hypothesize that a threshold concentration of IL-2 may be associated with increased regulatory T cells that dampen inflammation and thereby reduce the risk of HIV acquisition. We know that regulatory T cells, or Tregs, play a critical role in the establishment and maintenance of immune tolerance and homeostasis. Extensively studied in skeletal muscles and the gut, tissue Tregs have demonstrated marked adaptability to the maintenance of tissue integrity and function via the control of inflammation. However, few studies have examined Tregs in the female genital tract and their role with respect to genital inflammation and HIV acquisition. In this study, we sought to characterize cervical Tregs and to correlate these to socio-demographic variables and inflammation as a surrogate of HIV susceptibility. 68 women accessing treatment and care at the Sex Workers Outreach Program Clinic in Nairobi City were enrolled in this study. We collected cervicovaginal secretions used via menstrual cups for cytokine and microbiome analysis. We also collected paired cytobrushes and blood specimen for isolation of cervical and peripheral blood mononuclear cells. We then performed flow cytometry on freshly isolated cells and characterized our Tregs as CD4 positive, CD127 low, CD25 high, and separately gated these on FOXP3. We also characterized HIV target cells Th17 as CD4 positive, CCR6 high, CD161 high. In our study population, the median age of the women enrolled was 28 years, and the median duration of sex work was 3 years. 55% of the women reported having used PrEP, and 48% reported the use of contraceptives. 42% of these women reported the use of DMPA as their preferred contraceptive. Looking at socio-demographic variables and Tregs, we found that endocervical Tregs inversely correlated with the duration of sex work as shown in the top left figure, and this was also recapitulated in blood. We also found that the number of sex acts with repeat clients inversely correlated with Tregs in the cervix as shown in the top right figure. Furthermore, the history of stillbirth, miscarriage, or spontaneous abortion was associated with lower Tregs compared to live births. On interrogation of the phenotypic distribution of Tregs in blood and cervix, we found a higher proportion of Tregs in the cervix compared to blood, and that most of these Tregs express FOXP3 with no difference in proportions between the two compartments. Furthermore, we found that the proportions of Tregs correlated in the blood and the cervix. Cytokine analysis of cervicovaginal secretions revealed that 24% of the women had inflammation as defined by the expression of 3 out of 8 cytokines. Additionally, a significantly higher proportion of Tregs was observed among women without inflammation compared to those who had inflammation in their cervix. We have previously shown that inflammation is associated with increased HIV target cells in the cervix. So we next examined the relationship between Tregs and HIV target cells and found that the proportion of Tregs correlated inversely with the absolute CD4 and Th17 T cell counts, indicating that Tregs may be contributing to the regulation of HIV target cells in the female genital tract. In summary, we have successfully characterized cervicovaginal Tregs from cytobrushes and shown that the number of sex acts with repeat but not casual clients correlated inversely with Tregs. We've also shown that history of stillbirth, miscarriage, or spontaneous abortion was associated with lower Tregs in the cervix. Although this data is not shown in this presentation, we also observed trends towards lower Tregs in women who reported the use of DMPA as a method of contraception compared to those who used other methods such as oral pills and implants. We also show that Tregs were higher in the cervix compared to blood, but with similar expression of FOXP3. Women with inflammation had lower Tregs compared to those without inflammation in the cervix, and finally, we show that Tregs inversely correlated with HIV target cells in the cervix. In conclusion, our data provides preliminary evidence that Tregs could modulate inflammation and recruitment of HIV target cells in the female genital tract. Therefore, it is critical to better understand the mechanisms that regulate inflammation and its pathogenic effects, which might have a major impact on reducing the risk of HIV acquisition. I'll end by acknowledging all our study participants from SWAP Clinic, the members of the McKinnon Lab in that very colorful picture, our colleagues at the University of Nairobi, and the various funding agencies for their generous support. I thank you for listening, and I'll be glad to take any questions.